Hello, I'm Dr. David McNamara. I'm one of the infectious disease physicians here at Gundersen Health System in La Crosse. This is our third episode of our COVID-19 podcast series, and today I will be talking about vaccines for COVID-19. And I want to talk a bit about some of the hopes and expectations that we have for vaccines. But just to start, I do want to talk a little bit about the nomenclature. So you may hear words coronavirus, COVID-19, those type of terms. And just to clarify, COVID-19, that's the disease caused by this new novel coronavirus that we call SARS-CoV-2. In this talk, you may hear me refer to this as SARS-CoV-2, and that's to differentiate it from the original coronavirus that was causing SARS back in 2003. We've now designated that as SARS-CoV-1. And areas that I'll touch on, I want to talk a bit about some of the complexities of vaccine development, some of the three of the vaccine candidates that we have, some questions about safety and efficacy, and then finally touch a bit on distribution of a vaccine. And so to back up, I want to talk a little bit about complexities of vaccine development. And normally, vaccine development here in the U.S. and worldwide, it's actually quite a lengthy process. Typically takes about 15 to 20 years to bring a new vaccine product to market. You may have heard the term Operation Warp Speed, and that's a U.S. government program that's directing a strategic approach to try and develop a vaccine for SARS-CoV-2 and helping with planning and resource allocation. What is very different about Operation Warp Speed is that vaccine development processes are happening in parallel, not sequentially, which is the usual operation that industry would use in developing a vaccine. There's been a huge influx of U.S. federal government resources to fund very large trials involving 30 to 40,000 persons that have been done to try and adequately power studies to determine the efficacy and also evaluate initial safety data. This has shortened the vaccine development time to about 12 to 18 months, which is really, it's tremendously remarkable compared with our normal expectations for vaccine development of 15 to 20 years. A key aspect of this is that the program has assumed that vaccine candidates will be successful, and they've already started large-scale manufacturing, which is underway now. To do that, U.S. taxpayers, that's me and you, we have assumed about a $30 billion financial risk for vaccine development. So we're paying the vaccine industry to develop and produce vaccines prior to efficacy and safety being known. So the risk of this has been transferred from industry to the U.S. government and to us as taxpayers. The goal has been to produce and deliver a successful vaccine by January 2021. Now I'd like to talk about several of the vaccines that are in development that we've all heard about in the media in the last few weeks. There are 6 vaccines that are currently in development in the U.S., and I'll talk about 3 of them. Some of the background of the biology of COVID-19 and SARS coronavirus 2 is that its ability to transmit while pre-symptomatic and asymptomatic makes the virus very challenging to control respiratory transmission in the wider population. In addition, this virus pandemic has really unmasked very difficult and long entrenched racial, ethnic, and economic disparities in the U.S. and internationally, and vaccine distribution plans and testing and trials need to take that into account as well. Some of the challenges with the vaccine development, it's really been quite difficult without a very detailed knowledge of the biology and epidemiology of SARS-CoV-2. In particular, our knowledge and understanding of immunity to SARS-CoV-2 is incomplete, which makes developing a vaccine challenging. We don't really understand fully the antibody correlates of protection, and also we do not know the duration of immunity after natural infection or immunization. So that throws a lot of questions into developing vaccination plans or infection control in the wider population after people have had natural SARS-CoV-2 infection. And in part, we know that of the normal circulating coronaviruses, which can cause the common cold, immunity seems to be somewhat short-lived. So after we get those infections, we don't have permanent, long-lasting immunity like we would typically associate with many viral infections, such as varicella or chickenpox or the measles virus, those sorts of infections where after we're vaccinated or we have natural infection, we assume that we have very long-lived lifetime immunity. Coronaviruses that we know of don't seem to work that way. So this is a challenging aspect of this. Fortunately, we've had quite a bit of prior development work on vaccines for the SARS coronavirus 1, which we had an outbreak in 2003 of, and also MERS, or the Middle Eastern Respiratory Syndrome coronavirus. There was some work on vaccines for those infections, which has given us a bit of a foundation to work on for developing a vaccine for SARS-CoV-2. And also, we have some new vaccine technologies, mRNA vaccines. We have had development in the last few years of experimental mRNA vaccines for influenza and Zika virus, none of which have been studied in large-scale trials or rolled out into use in the population, but some of that development work was also an important foundation for scientists to build on during this present pandemic. The vaccine target for both SARS-CoV-1, MERS, and now a SARS-CoV-2 vaccine is the spike protein. And that spike protein allows the SARS-CoV-2, the coronavirus, to bind to the ACE2 receptor on human host cells. That initiates fusion of the virus with the host cell. The goal of vaccine production has been to make the human host develop an antibody to that spike protein, which would prevent virus fusion to the host cell and prevent infection of the human host. Now I'd like to touch on several specific vaccines in development. We've had some media reports in the last few weeks which have provided us with quite a bit of optimism and hope that there will be effective vaccines to help end this pandemic. And I want to touch upon 3 vaccines. 2 of them are the mRNA vaccines, and one of them is an adenovirus vector vaccine. In total, there are 6 vaccines in development for the U.S. market, including 2 of them using the older, sort of tried and true, well-developed technology of a protein vaccine with an adjuvant to help boost immune response. Of the mRNA vaccines, currently there are no licensed mRNA vaccines. So this really is a new technology. There are prior experimental and development work that's been done on mRNA vaccines against influenza and Zika virus, but those were not further developed or marketed. The experimental work showed that the mRNA vaccines, we think, are likely to be a very successful technology, but especially in the influenza virus vaccine marketplace, they were not felt to have real good market potential, so they weren't developed for FDA approval. One advantage of mRNA vaccines are that they can be very rapid to manufacture. So in a pandemic situation, such as if we had a very virulent pandemic influenza, or in the current COVID-19 pandemic, the speed with which scientists could develop and manufacture a vaccine may be an especially helpful aspect of these vaccines. To develop an mRNA vaccine, the scientists need to know the genetic sequence of the target protein. Then the manufacturer uses recombinant DNA technology to rapidly make many mRNA transcripts. Those mRNA or messenger RNA transcripts that would encode a protein of interest, which in this case is the SARS-CoV-2 viral spike protein. Those are encapsulated in a tiny lipid particle. They can be much less expensive and faster than traditional vaccines to produce. And in addition, they may be much safer, as they certainly do not use infectious particles. And they're really only introducing mRNA transcripts for one of the many proteins that a virus normally would produce. The mRNA transcripts in their lipid particles are then injected into a human, taken up in human cells, and the human cells use those mRNA transcripts to manufacture the viral protein. That foreign viral protein is secreted from our cells, and our immune system will mount immune response to that foreign protein. So this is really a new and very different technology than we've ever used before on a large scale for vaccination. Part of the downside or the challenges with mRNA vaccines are that we know they need multiple doses. So unfortunately, we can't give a single one-time dose. We're going to need to use 2 doses for these vaccines. Also, they have requirements for cold chain storage and distribution in very cold freezer temperatures. This makes distribution plans quite challenging. Also, we know that these vaccines tend to be reactogenic, which means they commonly, especially after that second dose, will give mild, self-limited inflammatory side effects, including fever, malaise, inflammation, and pain around the injection site, particularly after the second dose. So that's something we will need to communicate to our patients and the population, in a sense, to try and set expectations for these vaccines. 2 mRNA vaccines have been reported so far. The first is one produced by Pfizer and BioNTech, and that has had 95% efficacy in a 43,000-person trial. It's given in 2 doses, 21 days apart, with storage at -70 centigrade, stable for 5 days at refrigeration temperature. The cost of that vaccine is estimated to be about $20 per dose. The other mRNA vaccine that we've heard a lot about recently is by a company called Moderna, and that has been reported to have 94.5% efficacy, again given in 2 doses, this time 28 days apart, with storage at -20 centigrade, stable for 30 days at refrigeration temperature, with a cost of about $35 per dose. Certainly, the cold chain storage for both of these vaccines is a challenge with distribution, especially for the Pfizer vaccine. But overall, it appears that using an mRNA vaccine encoding that viral spike protein has been a successful endeavor to markedly reduce the amount of PCR positive confirmed infections in that study population. A third vaccine that we've heard a bit about in the last week or two has been an adenovirus vector vaccine produced by AstraZeneca and Oxford University. This is using a adenovirus from a chimpanzee population to deliver genes encoding the viral spike protein to human cells. And this has been used as a viral vaccine vector once before in a new adenovirus vaccine, which has just been approved against Ebola virus. So we do have some limited experience with adenovirus vector vaccines in the last several years in development. This also is a 2-dose series. It would require storage at refrigerator temperatures for up to 6 months, and the cost about $3 to $4 per dose. A huge advantage of this vaccine, if it's effective, could be its distribution requirements for storage are much easier and much more feasible, particularly in low resource settings. We don't have really detailed knowledge of this vaccine yet. The only knowledge we've had has been in press releases from AstraZeneca. So the data has not been peer reviewed and really we're awaiting that and is to be reviewed. There were some questions about the trial design. It appeared to have much more effectiveness in a subset of 2,700 persons who were accidentally given a half dose of the initial vaccine, then a full dose of for the second vaccine, they got the full dose. So that raised a lot of questions in scientists' and reviewers' mind. Some questions about the SARS-CoV-2 vaccines mainly center around questions of safety and efficacy. And part of the challenges of knowing this and evaluating this really has to do with that COVID-19 is a new disease, and the spectrum of illness and the disease manifestations are really now only becoming clearer the longer we've been living with this. So rare complications of natural SARS-CoV-2 infection and their sequelae, those are harder to recognize without extensive experience. The current trials of the vaccine have been designed to provide 2 months of post-vaccine surveillance to detect common adverse effects prior to FDA review. But certainly going forward, we know that when we expand the vaccination to much larger populations, that post-introduction safety surveillance to detect rare complications will be especially important, with ongoing vigilance for that. Some of these vaccines right now, the Moderna vaccine, have been submitted to the FDA for emergency use authorization. And the criteria for that the FDA uses are certainly not nearly as rigorous as the usual FDA approval process. The standard really is that they determine that a product may be effective and that benefits are likely to outweigh the risks. So that's, in a sense, that's a fairly low standard compared to their normal FDA approval. The FDA has set a threshold of 50% effectiveness for a SARS-CoV-2 vaccine and has said they will only approve a vaccine with substantial evidence of safety and efficacy. One aspect of the trials and the science that we have that I would like to highlight has to do with effectiveness of a vaccine. And it's important to keep in mind the trial endpoints that we have really have been designed to measure whether or not the vaccine prevents PCR-confirmed illness in a trial participant. And that is somewhat different than preventing transmission of infection in the wider population. And it's important to keep that in mind because we all have a hope and an aspiration that introduction of a vaccine will allow us to relax other infection control measures such as social distancing or mask wearing. But it's important to understand the science is not yet real clear on the ability of vaccine candidates to reduce transmission within a wider population because the trials were really designed to assess whether or not the vaccine candidate would prevent PCR positive confirmed illness in the trial participation. One other thing that I want to highlight or talk about is the concept of herd immunity. And we've often heard it said, oh, we're really hoping to have herd immunity within a population to try and help reduce ongoing COVID-19 and SARS-CoV-2 transmission. For many serious viral infections, achieving herd immunity via natural infection is very difficult to achieve. That would require a significant population burden of morbidity and mortality. So this coronavirus, like many other infections, it's not really realistic to expect we're going to get herd immunity through natural infection. The hope of the vaccine would be that this could help give us herd immunity by reducing transmission within our population. But again, it's important to know that the science is not yet there in terms of supporting that. The last thing I want to talk about briefly is just some thoughts on a distribution plan. We will be in a supply constraint situation at first, and the logistics of delivering this vaccine to larger populations are going to be very challenging. The CDC and ACIP, or Advisory Committee on Immunization Practices, they are formulating vaccine distribution plans and prioritization right now. And currently it looks like the top three groups that will be targeted for vaccine immunization initially are going to be healthcare workers and residents of long-term care, then essential workers, including police, firefighters, educators, those involved with transportation, food, and agriculture, and then third, adults with high-risk medical conditions and those who are over 65 years old. As healthcare providers, we can expect a lot of questions from our patients regarding these vaccines. So we need to educate ourselves on available vaccines, particularly the efficacy and safety data, because I think those are going to be the main questions we will encounter from our patients. Some patients will really clamor for the vaccine. Many patients will probably have significant vaccine hesitancy, and there will be significant misinformation available for patients to read. So I think they will be looking to us for guidance, and we need to be prepared and educated on responses and information that we can advise our patients on. So in conclusion, we really do have a very robust pipeline of good vaccine candidates. The efficacy and initial safety data, particularly in regards to the mRNA vaccines, so far is quite good. The studies have been very large and well-powered to reach efficacy targets. So those releases of data and peer-reviewed publications that have come out about those have really been the first thing that have given the medical community some optimism towards the future in dealing with the coronavirus pandemic. And Gundersen is planning right now for distribution of vaccines through our partners in the state of Wisconsin for initial vaccine administration to priority groups. Thanks for listening this week. Tune in next week for another COVID-19 update.